Inside you, right this second, there are literally trillions of microscopic machines at work. We call them proteins. They're the ones building your muscles, digesting your food, even fighting off diseases. But here's the crazy part. How does a simple string of biological code, a blueprint really, create these incredibly complex and powerful machines? Well, today, we're going to pull back the curtain and explore the 4 secret levels of protein architecture.
And that, right there, is the central puzzle we're going to tackle today. I mean, how does a simple, one-dimensional chain of molecules figure out how to fold itself into this unbelievably complex, three-dimensional machine? It's definitely not random. The answer lies in this really elegant 4-level hierarchy of structure, where each level just builds right on top of the last one.
All right, so here's our game plan. We're going to start with the basic code, then see how it makes its first folds, forms its final 3D shape, and even assembles into bigger complexes. After that, we'll connect structure to function and wrap it up with a review. Let's jump in.
All right, our journey starts at the most fundamental level, the primary structure. Think of this as the protein's unique identity, written in the language of amino acids.
So, the crucial thing to remember here is that primary structure is just the specific sequence of amino acids, all linked together by these super strong peptide bonds. It's kind of like a string of letters that spells out a very precise instruction, directly translating the message from our DNA into the protein's starting form.
Let's look at insulin, for example. It's actually made of 2 separate chains, an A chain and a B chain, and the exact order of amino acids you see here, it is absolutely critical. Just one single mistake in this sequence, one wrong amino acid, and you can end up with a protein that doesn't work at all, which is the cause of some types of diabetes. So yeah, this shows it is definitely not random. It's a super precise blueprint. Okay, so that linear chain, it doesn't just stay straight. Almost immediately, it starts to fold into these regular, repeating patterns. And this, my friends, is the secondary level of structure, the very first step toward creating an actual 3-dimensional object. And at this level, 2 main patterns really dominate. On the left, you've got this elegant coil called the alpha helix, and on the right, we have this folded, kind of accordion-like structure called the beta-pleated sheet. Let's take a closer look at how each one is formed. So what's fascinating is what holds this helix together. It's not the unique side chains of the amino acids doing the work here. Nope. It's the repeating backbone itself. See, a hydrogen bond forms between an oxygen on one part of the backbone and a hydrogen on another part, exactly 4 amino acids down the line. It's this super regular, repeating pattern of bonds that coils the chain up into this tight, very stable helix.
And the geometry here is unbelievably precise. It's a right-handed coil, and it has exactly 3.6 amino acids in every single full turn. But here's the really important part. Notice how all the R groups, the unique side chains that make each amino acid different, they all stick out from the coil. And this is absolutely key because it leaves them totally free to form other bonds later on, which is what's going to determine the protein's final 3D shape.
Okay, now for the beta sheet. So instead of a coil, here we've got segments of the chain lining up side by side. In what we call the anti-parallel version, the chains are running in opposite directions. And that alignment, it's perfect for forming these straight linear hydrogen bonds between the backbones. And just think about it. A straight bridge is way stronger than a curved one, right? Well, it's the same idea here. These linear bonds create an incredibly stable pleated sheet structure.
Now, the chains can also line up going in the same direction, and we call that a parallel beta sheet. But if you look really closely at the diagram, you'll see the hydrogen bonds are now angled instead of straight. And that angled geometry makes them just a little bit weaker and less stable than their anti-parallel cousins. It might seem like a subtle difference, but trust me, it has a big impact on the overall stability of the protein.
Okay, so now we've got our coils and sheets. What's next? Well, the next step is to take all of these secondary structures and all the loops and turns that connect them and fold them all up into one final, compact, and functional 3D shape. And that is the tertiary structure.
And this image of myoglobin is a brilliant illustration of that concept. You can clearly see the alpha helices. They're the pink ribbons, folding and packing on top of each other to create this super compact globular protein. And it's this final, specific shape that lets myoglobin do its very important job, carrying oxygen in our muscles.
So what locks this incredibly intricate shape in place? Well, it all comes back to those R groups we saw sticking out earlier. They interact in 4 main ways. You've got weaker attractions like hydrogen bonds, Then you have these really strong permanent staples called disulfide bridges. You also get these magnetic-like attractions between positive and negative charges. We call those ionic interactions. And finally, there's this really powerful organizing principle. The hydrophobic or water-fearing R groups all huddle together in the protein's core to get away from the water surrounding them.
Let's circle back to insulin for a second. Remember those two separate chains? Well, the thing that locks them together in their exact orientation are these disulfide bridges. These are super strong covalent bonds between sulfur atoms on specific amino acids called cysteine. They really do act like molecular staples, just permanently locking the structure right where it needs to be. But wait, we're not done yet, because sometimes just one folded polypeptide chain isn't enough to get the job done. This brings us to the final level of protein architecture, quaternary structure. This is when multiple, separate polypeptide chains, each with its own fully formed tertiary structure, come together to form one big functional complex.
And hemoglobin, the protein that carries oxygen in our blood, is the absolute classic example of this. See, it isn't just one chain. It's a precisely assembled complex of 4 separate proteins, 2 alpha subunits and 2 beta subunits. This assembly is quaternary structure, and it's what allows hemoglobin to be so good at its job, skillfully picking up oxygen in the lungs and then releasing it in the tissues. That's a task one subunit just couldn't do by itself.
Okay, we've gone through all 4 levels of structure. So now let's connect the dots and see how this architecture directly determines what a protein actually does in the cell. Because when it comes to proteins, shape is everything.
So, broadly speaking, proteins fall into 2 main structural classes. First, you've got fibrous proteins, like the collagen in your skin. Their strength really comes from their secondary structure, these long, repeating helices or sheets that form insoluble fibers. They're perfect for building things. Then you have globular proteins, like enzymes or our old friend insulin. For these guys, the complex tertiary structure is the key, folding them into these compact, water-soluble shapes that can travel around the body and perform all sorts of chemical tasks.
And this amazing structural diversity allows for an incredible range of functions. I mean, proteins can be structural building blocks like collagen. They can be biological catalysts like enzymes, chemical messengers like hormones, little delivery trucks like hemoglobin, or even bodyguards like antibodies. The key takeaway here is simple. For every unique job in the body, there is a unique and perfectly evolved 3-dimensional shape to do it. All right, we have covered a lot of ground. So let's do a quick review of the 4 levels of protein architecture one last time, just to make sure these concepts really stick. Okay, let's put it all together. At the primary level, you have the simple amino acid sequence, held together by peptide bonds. Then, at the secondary level, you get the first local folding, your alpha helices and beta sheets, and that's all stabilized by hydrogen bonds in the backbone. The tertiary structure is the overall 3D shape of one single polypeptide, locked in place by all those R group interactions, disulfide, ionic, H bonds, and hydrophobic forces. And finally, quaternary structure is the arrangement of multiple subunits, using those very same forces to hold the whole complex together. And this single image really captures our entire journey perfectly, doesn't it? We start with that primary sequence, just a simple string. That string then folds into secondary structures. Those structures then pack together to create a tertiary shape. And finally, multiple shapes can assemble into a big quaternary complex. It's truly a hierarchical masterpiece of molecular engineering. Which leaves us with one final and pretty profound mystery. A single protein chain has an almost unimaginable number of ways it could theoretically fold. So, how does it find its single, correct, functional shape in just a fraction of a second every single time? Well, solving that incredible navigational puzzle, that's a story for another time.